Hello everyone and welcome to the lecture series on molecular dynamics and simulation. This is Rohan Mishram, and in our previous video I have just briefed you on as to how motions happen in your protein, right? And why is that we need to study this molecular dynamics, or you can say the dynamics that happen in your protein. Okay, then we discussed like what are the two types of motion that might happen in your protein. We have classified the motion into fast motion and slow motion, and I have just introduced you to what are the different techniques that exist that are used to monitor this fast motion and slow motions, right? Then we discussed how do we describe our system in form of this particle based description, right? And we have also discussed as to how exactly the systems are supposed to be modeled for your molecular dynamics and simulation. What is to be included, where exactly do you need to stop, and what is to be considered, what is not to be considered, those sort of stuff. Okay, and I have introduced you with a very few elementary concept in molecular dynamics, like you need to run your simulation in say some limited period of time, and there are trajectories that are supposed to be generated by each simulation method, and these trajectories can be formed of various frames that are captured one after another. Okay, so now loaded with this basic knowledge, today we are going to learn some few more basic concepts that would be very useful for you in your subsequent lectures in molecular dynamics. Okay, so what we have discussed last time was that you might end up with a trajectory that can be formed of various frames, and these frames would correspond to say some snapshot that you might have took along the course of your simulation. Okay, so now to continue your discussion, you can say that you can sample the relevant properties that can be averaged over the entire trajectory. Okay, so let me explain you what exactly I meant when I just said that. Okay, say now you have a trajectory, and now what you can do is that you can plot the energy values over the entire trajectory. Okay, so this graph might look something like this. Say for example, if you have this trajectory that you have plotted. Okay, well, say for example, this is time on x-axis, and you can plot conformation of your protein, or say energy value of your protein, okay, on the y-axis. Okay, so this is how your plot might look like, right? So this is how the energy or conformation of your protein might change over time. Okay, so say for example, in this simulation you can now select some states at some time interval. Let us continue with our previous example of 50 nanosecond simulation, right? And say at interval of every 50 picoseconds your protein will have different conformation. Just to begin with, say at this 50th picosecond of time, your protein might have this conformation. Okay, that might look like this, right? Then at 100 picosecond of time, your protein might look something like this. Okay, then when your simulation progresses forward at 150th picosecond, your protein might look something like this. Okay, it might have this conformation. Well, say for over here at say 200 picosecond, your protein conformation might be something like this, so on and so forth, right, till you reach the so on and so forth till you reach the final nth conformation, right, where your protein might, will say for example, look something like this. Okay, so in this way, throughout the trajectory the conformation of your protein might change, and this is how your protein might look like in your trajectory. Okay, now at these specific this specified interval of time, what you have done is that you have saved the snapshot of your system. Okay, and now you might be interested in, say, some interesting property of your system. Okay, say for example, you might be interested to monitor how free energy changed over this simulation. Okay, therefore, for every snapshot that you have captured, you can calculate the interested property, right? In our case it is free energy, and therefore at, we'll say, 50th picosecond of a state, the value of free energy is, say, some x1, right? At 100th picosecond the value of your property of interest, that is the value of free energy, would be, say, x2, right? And at 150th picosecond the value of energy would be x3. Okay, at 200 picosecond the value of free energy would be, say for example, x4, so on and so forth till the nth picosecond we will be having the free energy value, say, xn. Okay, so this is what you can do. So in this way we say that we are sampling, what we say, we are sampling the structures that are present in your trajectory. Whatever properties you are interested in, you can average up those property value and say that the average free energy change in this simulation was, say, some xxx kilocalorie per mole. So that is what exactly we are going to do. We are going to average, that is what we are going to do is that we are going to add x1 to x2 to x3 to x4, we are going to add till xn and divide it by the number of frames that you have saved. Okay, so and whatever average free energy that you calculate, you can say that this is the overall free energy of my protein ligand system over the specified given interval of time. Okay, so in short what I want to convey is that some relevant properties of each and every frame can be calculated and you can average it over the entire trajectory. Well, this is, sampling and calculating and averaging things in molecular dynamics and simulation will be, the most frequent task you might do when you perform such MD simulations. Okay, so this is what I really mean. I want you to get introduced with the concept of sampling. Now there are various sampling methods, like, we'll say for example, you can have umbrella sampling, okay, and various other sampling methods that we might I might prepare a different video for how do we do different type of sampling in molecular dynamics and simulation. Okay, so now I hope you are comfortable with the concept of trajectory sampling, okay, or sampling that happens in your molecular dynamics and simulation. Okay, so now let us move on to the next very important topic, like what are different type of molecular simulation that you can perform. Okay, so to be very brief, there are two main categories of molecular simulation, and these two main categories are based on how you propagate your system in time. If you propagate your system deterministically, then the molecular simulation you are performing is called as molecular dynamics simulation. On the other hand, if you use probabilistic method to propagate the system, then we say that you are conducting Monte Carlo simulation. So molecular simulation can either be deterministic or they can be probabilistic. By probabilistic, I mean that the simulation, the propagation is based on, say, some probability principles. We are going to deal with these two concepts in more details in upcoming few slides. Okay, so let me try to just summarize it as to what are the exact basic difference between such MD simulation and Monte Carlo simulations. Okay, so the very first difference between this MD simulation and Monte Carlo simulation lies in as per how they are used, right? As mentioned here, as you can see over here, these MD simulation are used to investigate structural, dynamic and thermodynamic properties of your system. Okay, you might want to use this MD simulation to observe how the system has evolved. You might be able to analyze dynamics of the system, as well as you will be enabled to figure out the thermodynamic properties of your system. On the other hand, over here, Monte Carlo simulations are used to access the structure, used to address the structural and thermodynamic properties of the system, but they cannot be used to infer the dynamic properties of the system, because the concept of time is not strictly followed in MC trajectories. And why is that? Well, we will come to this point in a little while. Just hold on to that question. Okay, so in short, from this point, you can say as to how you can use this MD simulation. If you want to monitor structural, dynamic as well as thermodynamic properties, then you should be conducting MD simulation. But somehow, if you are not interested in dynamic properties, but you are interested in structural and thermodynamic properties of the system, then you are supposed to use this MC simulation. Okay, the second difference lies in the aforementioned fact that deterministic rules are used to generate new confirmation from the present confirmation. Okay, so what happens in molecular simulation is that every time when you begin with, say, some state of your system, we'll say for example this is the initial state of your system. Okay, so every time you begin with some state of the system, and subsequently what you do, you would try to calculate the state of the same system after, say, some delta t time has passed. And what exactly happens in this delta t time, what will be the new state of my system after delta t time has passed. Okay, this is exactly what we do in molecular simulation, right? So whatever new confirmation that is generated after this time delta t, we use some deterministic rule to get this thing done when we are talking about this MD simulation. Now, if you are using these deterministic rules in finding out what your new confirmation might look like, then my friends, we say that we are performing MD simulation. On the contrary, in MC simulations, some probabilistic rules are used to generate this new confirmation from the present confirmation. Right? So here is the second difference. Right? The third difference may lie in practical perspective, right? So let me explain you what I mean by that. So this deterministic rules based simulation or simply MD simulation methods are more ideal when you have a folded state of a protein. That is, you already know beforehand the protein is folded, and this is how the folded state of my protein might look like. Okay. And your objective in this situation is to monitor how structural changes happen in the protein over the given time span. Right? You already know that whatever protein that I have with me, the initial state of the protein is already folded. Okay. And your objective is to monitor what structural changes might happen in my protein over the given period of time span. Right? Then this MD simulation is the method you would be looking for, my friends. Okay. Using MC simulation, right, will be more ideal when you have to start out with something in random state. Okay. If you start out with, say, some random state of the protein, which you know is not the folded state of your protein. If you begin with a random state of your protein, okay, and and your objective is to find out as to what new conformation this random state might acquire. Okay. Over here in MD simulation, you begin with the folded state of protein, and all you want to do is to monitor how this protein's conformation might change. In case of MC simulation, you don't begin with a folded state of protein, and your objective is to figure out as to what might be the next conformation of my given protein. Okay, so objective is to find out as to what new conformation this random state might acquire. Okay, so this is the basic difference between MD simulation and Monte Carlo simulation. Okay, now the next and fourth difference lies with the concept of time, and if or not it is handled during the molecular simulation. For example, your MD simulation will generate something called as the continuous dynamic trajectory. Okay, while the trajectory that are obtained in Monte Carlo simulation are not continuous dynamic trajectory. Okay, these are not continuous dynamic trajectory. You cannot consider them as continuous trajectories. Right? We will come to this point just in a while. Okay, so uh, coming to the fifth point of difference, the trajectories that are generated during MD simulation includes the concept of time. This is what is very important, that whatever trajectories are generated in MD simulation, that includes the concept of time. We know that the simulation start at some point, and after, say, some time, it would end at some point. Okay, by that I mean that the frames that are captured in the trajectory will appear in the same order in time. Okay, so this is exactly not followed in case of Monte Carlo simulation. Okay, in Monte Carlo simulation, the trajectories do not include the concept of time. So whatever frames you visualize in MC Monte Carlo simulations, that would represent an ensemble of configuration that could be structurally sampled. Okay, in short, in simpler words, the frames that you will see in Monte Carlo trajectory are just collection of state of a protein that are thermodynamically and kinetically accessible. Okay, let me maybe show you a Monte Carlo trajectory, and let us compare it with MD simulation trajectory. Okay, so whatever you have seen so far, the trajectory that I have shown you in my previous video, those were for molecular dynamics and simulation. Now I am going to show you the trajectory that is derived from this Monte Carlo simulation. Well, let us view this trajectory. Okay, so here, let us view this trajectory as how the state of your system changes in this trajectory one after another. Okay, you might see various frames that are being shown over here, right? And here, this trajectory, it might appear the same as in MD simulation, but there is one basic difference, my friends. The difference being the frames that that you have seen in case of MD simulation are happening one after another as they appear in the trajectory. On the other hand, you know, on the other hand, uh, you might observe sudden shift in secondary structure in this Monte Carlo simulation trajectory. For example, in one frame, you might see a strand or a helix, and in next frame, you might not see it. Okay, let us see this video once more if you observe this fact. Okay, well, say for example, there is helix over here, right? And there is helix, suddenly a helix appears over here, right? And suddenly it would disappear over here, right? So this observation indicate, or this observation demonstrate, that whatever frames that you have just seen in this Monte Carlo simulation trajectory, these frames that you see that are observable in this MC trajectory are simply states of that protein that it can acquire, but it is not necessary that the protein would acquire these states in the same order as they appear in the trajectory. Okay, well, now the path to acquire these states, as you can see over here, the path to acquire these states can be different. Okay, and hence we say that time is not exactly followed in Monte Carlo simulation. Okay, the states that are these are the states that are accessible to this protein, but not necessarily in the same order as it appears in this trajectory or as they appear in this movie. It can acquire the last frame that you see in trajectory somewhere middle, or it can happen the otherwise. Okay, therefore frames in Monte Carlo trajectories can be termed as just ensemble, that is a collection of structural states irrespective to the time at which it might be achieved. Okay, so this is the basic difference between the trajectories that you obtain from Monte Carlo simulation as compared to those that you obtain from MD simulation. Okay, so the frames that you would see in molecular dynamics simulation that I have already shown you using that VMD software, those are the state of that protein that would happen real time. The protein would change its conformation as you can see in the trajectory. On the other hand, in Monte Carlo simulation, it is not necessary that the shift or changes in frames that you see in trajectory might happen in the same order as they appear in your movie. Okay, so there can be, it can happen that your protein might acquire some state that is shown later in your trajectory. It can already acquire that state at the initial state when it really folds on. Okay, so now let us deal with this question: when to do what? So based on whatever discussion we have done so far, Well, let us simplify and answer this practical question as to what methodology should I adopt. Okay, when should I use this Monte Carlo simulation and when should I use this molecular dynamics simulation? Okay, so when to aim to so for example, uh, when you aim to monitor structural changes in already folded protein, you should go for MD simulation, right? If your objective is to figure out as to what happens to my protein, which I know is already folded, then you should go for MD simulation. On the other hand, the very first feature of Monte Carlo simulation is that it allows you to pick up some random move, and it's not necessary here to know where to start and where to end. Okay, so this feature might be more suitable when predicting the structure of a protein. Why? Because we really don't know where exactly your protein starts to fold and where it stops, right? And Monte Carlo exactly allows you to do, it would allow you to try some random things and see what really works after taking those random moves, right? Since Monte Carlo simulations are based on a lot of probabilistic concept of propagating the system and a lot of randomization is involved in such Monte Carlo simulation, it is therefore included in special class of algorithm called as stochastic algorithm. Okay, as it involves lot of probabilistic and lot of randomness is involved while you propagate your system. Okay, so therefore these ab initio structure prediction usually prefer to adopt a Monte Carlo simulation. Okay, so now discussing the concept of Monte Carlo simulation is not in scope of these video lectures, right? Here we will focus our attention on MD simulation. If you want to know more details of the Monte Carlo methodology and the algorithms that are implemented in Monte Carlo methodology, well, you are advised to watch my ab initio structure prediction video. So here onwards, we will only talk about these MD simulations, right? So, let me summarize what exactly we have learned in this particular session, right? What we have learned is that what are what exactly is sampling, right? And what exactly do we mean when we say that we are sampling the trajectories? Okay, what you exactly do when you sample the trajectory. Then I have tried to classify molecular dynamics and simulation, these molecular simulations into two broad categories: deterministic based simulation and probabilistic based simulation, right? Deterministic based simulations are molecular dynamics simulation and probabilistic based simulations are Monte Carlo simulations, right? And we have kind of tried to differentiate between what happens in molecular dynamics and simulation and what happens in Monte Carlo simulations, right? And I have shown you the trajectory of a typical Monte Carlo simulation. Okay, and we have also discussed as to when you should adopt this Monte Carlo simulation and when you should adopt this molecular dynamics simulation. So, this is what we have done so far, right? So in our next part we would try to discuss some more basic aspect of molecular dynamics and simulation, right? So see you guys in our next video lecture.